Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
The present chapter, Establishment of Cultures, Protoplasts and Anther Culture, will be explained by the Dr. A. Veerbrahman, HOD, Assistant Professor, Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The present chapter, Protoplasts and Anther Culture, having learning outcomes. In this chapter, students will be able to be acquainted with the concept of highly in vitro process, assess the different methods of protoplasts and anther culture, determine the factors involved in protoplasts and anther culture, evaluate the mechanism of haploid embryogenesis, and reproduce the protocols for pilot scale operations. So especially in the first outcome of the concept of highly in vitro process. In vitro means within the laboratory or within the glass, the callus or aseptic cultures will be prepared. So in the present chapter, the protoplasts can be isolated, anthers will be isolated and aseptically transferred to the biosafety cabinets and will be inoculated on a required media. In the second outcome, that is protoplasts and anther, variety of methods, different methods available to culture the protoplasts and anther. The third objective, that is a factor. So what are the factors? That is media constituents, especially carbon source, nitrogen source, and growth regulator combinations, especially for protoplast culture, auxin-rich media, especially 2,4-D, proved to be very effective for upgrading the protoplasts. And for anther culture, it's an organogenesis. So that the combination of the combinations of the hormones will be reversed. Cytokinins and auxins will be reversed. Here, the cytokinin concentration will be enhanced and the auxin concentrations will be reduced. Okay, and the next outcome, learning outcome, that is the factor mechanisms, haploid embryos. So what is a haploid? Haploid is a cell or an organism containing a single set of organisms. Here embryogenesis, haploid embryogenesis, the embryo having a single set of chromosomes. So these protocols can be reproduced on a pilot scale operations in the laboratory or whenever the conditions are conducive to produce the same kind of or same lines for industrial purpose or a commercial purpose. So let us see the contents in the present chapter, Introduction to Plant Tissue Culture, that is protoplast and anther culture, protoplast isolation, methods involved in the isolation, and protoplast culture, variety of culture techniques, and regeneration of the protoplast. Anther culture, what is anther and a process, factors involved, applications of both the protoplast and anther culture. So let us see first introduction to the plant tissue culture, that is protoplast and anther culture. Plant tissue culture is a process of culturing any plant parts of choice from the next plant in a sterile environment using in vitro methods. So very efficient way to produce large number of identical plants rapidly. So within a space, less constraint, and arranging or preparing an artificial environment where the plant can easily sustain itself and can be produced to a scalable amount of the plantations, ensuring genetic consistency. Okay, so through the in vitro process, especially in protoplast culture, there is a chance of getting in genetic constituency, consistable genetic clones can be obtained, and it focuses on protoplast and anther culture, covering applications in research and industry. Also define protoplast culture, isolation, generation, regeneration, and finally it covers anther culture and factors affecting the success. Let us first dive protoplast isolation methods and considerations. So we have two kinds of, principally we have two methods, so that is mechanical methods and enzymatic methods. Mechanical methods generally involve manually slicing the plant tissues that releases the protoplast. It's a very less efficient. Whereas in case of an enzymatic method, usually utilization of the enzymes like cellulose, cellulase and pectinase, these digest cell walls, enzyme concentrations and incubation times must be optimized. Then only you will it will be obtained. It has been obtained right content. I mean, more profitable deals with the enzymatic method, though they are costly effective, so very useful and less laborious. So protoplasts are plant cells without cell walls. Isolation affects protoplast yield, plant species and enzyme purity matter. Let us see the protoplast culture techniques and regeneration. So first of all, for protoplast culture, culture media, cultural conditions, protoplast fusion are the main portions in the protoplast culture. Culture media, generally morasticae and spook or a B5 medium can be used to sustain the plant materials and vitamins, hormones essential for plant growth and development. The composition varies depending upon the plant species. So one species requires special conditions to sustain it themselves, to gives them more number of clones. So the cultural conditions comprising clean and sterile environment to minimize contamination. Generally 70% ethyl alcohol, surface sterilization, chemical sterilization, physical modes of sterilization can be applied to maintain or to attain the cleanliness, hygiene in the plant tissue culture practices. So typically involves controlled light, ambient temperature, that is 25 plus or minus 2 degrees centigrade is required, and adjusting the pH 5.5 is the optimal pH required for the protoplast culture, which is acceptable throughout the plant tissue culture operations. Protoplast fusion. The fusion can be achieved through the physical agents, either physical agents or chemical agents. Physical agents, gentle tapping of the protoplast isolated in a bovine sera, isotonic solution, or chemical methods, high calcium or a polyethylene glycol mediated method or electrofusion. These are very effective methods to get the right choice, protoplasts in hybrid mode. Okay, plants regenerate through callus formation, somatic embryogenesis and organogenesis are also used to generate the protoplasts. Among the chemical methods, high calcium, polyethylene glycol mediated and electrofusion methods. Polyethylene glycol method is the very choiced or prioritized method where less chances of contamination and shock to the protoplast and whereas in case of electrofusion there is a thermal shock whenever accidentally if the kinetic energy is increased by chance. Since the protoplasts are very sensitive, they may get the shock, so that the junctions between the two cells, between the two protoplasts will be damaged. So that is the problem with the electrofusion. Whereas in case of high calcium method, where it generates high acidic conditions, acidic medium, so that the media constituents may not be available for the growth and development of the protoplast into a callus regeneration. So, which is the optimal or the first step to get the plant regeneration. So these are the applications for the protoplast culture: genetic transformation, somatic hybridization, disease resistance. Genetic transformation, generally direct DNA uptake, enhances the traits. So this is very feasible with the protoplast, since they are naked, cell wall minus cells. That's why the DNA can easily be taken up into the cell. And somatic hybridization. So in this case, combines. Since somatic hybridization combines the traits, like the tomato is a good variety, was already prepared through the somatic hybridization. Somatic hybridization is nothing but hybridizing the somatic cells without cell wall, because the protoplasts can easily be under the stimulation either of the physical agents or a chemical agents. They can easily be share the cytoplasmic genes and the protoplasmic genes to get a new variety. And disease resistance. So protoplast culture screens for resistance at the cell level, how much the disease is there and how much the potential pesticides or fertilizers are taken up by the cells. So protoplasts aid metabolic studies. They enable crop improvement. They also allow genetic modification for hybridized. Now enter the anther culture process factors involved. Anther culture pre-treatment includes culture media, produces haploid plants. The best stage is the uninucleate microspore stage is required for the anther culture. And permanent pre-treatment includes the cold temperatures ranging from 4 to 10 degrees centigrade for 1 to 7 days is optimal condition. The culture media is usually MS Murashige and scope media is required. N6 also medium is also used for the anther culture. Plant growth regulators are also used to culture the anthers. Of course, since it is an organized culture, this is cytokinins are more required than the auxins, and the culture conditions required controlled light temperature, usually 25 to 28 degrees centigrade is required. The process and factors involved continuous anther culture technique. The technique involves the excising closed flower buds which have anthers containing uninucleate microspores. At this stage, microspores are most suitable for the induction of androgenesis. The excised flower buds are surface sterilized. Anthers are removed. Care should be taken to avoid injury to the anther walls or anthers cultured on agar solidified or a liquid media. Either of the solid agarized media or a suspension culture can support the anther culture. And the cultures are incubated at 24 to 27 degrees centigrade using light about 2000 lux for 14 hours per day. So this is a schematic view of the anther culture. First of all, bud is opened, anthers are collected, anthers cultured on nutrient agar medium. Then, which under the stimulation of the hormonal combinations, that is a high concentration of cytokinin and less concentration of auxins, that enhance the embryoids, since it is an organized culture. Of course, either reversing the auxins to cytokinins can also enhances the callus phase. But here the embryoids can directly give rise to haploid plantlets from the anthers. A Depending upon the plant species, it takes 3 to 8 weeks for pollen plantlets to regenerate from anthers. So this is a huge period, about a 2 months long period to get the plantlets from the anthers. Of course, care must be taken while you are any contamination, browning of the media or reddening of the media, acidic, high acid production because of the cultural conditions, abnormalities. After 1 month of incubation, the microspores either develop into embryos or calli emerging through the anther lobes. Subsequently, the calli can be transferred to regeneration medium. In some cases, some pollen grains develop into embryos, and other calli within the same anther can also be inoculated on the same conditions, that is MS media supplemented with a high concentration of the auxin, high concentration of the cytokinin to auxin rich. The pollen plantlets about with a height of 12 to 14 centimeters are hardened and then transport to the soil. So that is the hardening process. So let us see what are the factors involved in the anther culture. Since it is a very organized culture, activated charcoal, temperature, stays of the anther are the very important factors. So that determines the possibility success rate of the anther culture. So here, activated charcoal, in addition to the carbon source, has a stimulatory effect on the embryogenesis, and this has been observed in an anther culture of potato, rye, tobacco, etc. So generally 25 degrees centigrade is required for the establishing the plant tissue culture. Here is a very significant that the pre-treatment of at 3 to 10 degrees centigrade for 2 to 30 days, a very long period of the anthers to stimulate embryogenesis. So here the embryoids or embryonic cells get to be activated, and another stage of anther, usually anthers just before or immediately after pollen mitosis, are most suitable for the culture. And photoperiod and light intensity, anthers obtained from plant source grown at increased light intensity or short duration may yield more embryoids. Flowering time, anthers collected from flowers at initial transformation phase or optimum for culture. Age of the plant, usually unmatured plants are more appropriate for culture. The matured plantations may not give the proper anther culture. So that is one of the obstacle or problem with the anther culture. So let us see the applications of and future prospects of the both the protoplasts and anther culture. So basic cytogenetic research studies can be possible through both the protoplasts and anther culture, and genetic recombination in higher plants are more possible through the protoplast culture, since they are the cell minus cell wall minus cell constituents. They can easily mingle or mix each other and forms the callus very easily under minimum concentration of the auxin. And differentiation of single cells transformation into whole plants, either of the protoplasts or anther culture, can be possible. Study of pollen embryogenesis of higher plantations, because you have to the embryos must be screened before the anther culture has been initiated. So that is most possible with the anther culture. Verification of double haploids, whether homozygous or fertile, since throughout the plant tissue culture operations, homozygous plantations can more conveniently generated through both the practices, that is protoplasts and anther culture. So these are the references for the topic just explained, protoplast culture and anther culture. Thank you for your attention, and we'll meet you the next lecture, next chapter. Thank you very much.